Welcome to botany class. Let's move to the next topic, bacteriophage. We shall discuss the structure of bacteriophage and the multiplication of bacteriophage, that is life cycle of bacteriophage. So today's learning objectives are structure of bacteriophage and life cycle of bacteriophage. So first question is define bacteriophage. Viruses infecting bacteria are called as bacteriophages. Viruses infecting bacteria are called as bacteriophages. It literally means eaters of bacteria. So these bacteriophages are abundant in soil, sewage water, fruits, vegetables, and vegetables, even milk also, milk. So bacteriophages are abundant in soil, sewage, fruits, vegetables as well as milk. So let's see the structure of bacteriophage. Look at this diagram. This is the structure of T4 bacteriophage. The T4 bacteriophage, which consists of a head part, this is a hexagonal head, a head followed by a tail, the tail which consists of inner central core of sheath, and the head and the tail is connected by a collar. So the bacteriophages which consist of a head, tail which is connected by the head and the tail connected by a collar, and the base of this tail there are basal plate. This is the basal plate and tail fibers with spikes are present in it. Inside the hexagonal head, the head is made up of protein. So it consists of 2000 identical protein subunits. The hexagonal head is made up of 2000 identical protein subunits. Inside the head, hexagonal head, there is a genetic material, that is DNA, dsDNA, which is called double stranded DNA, dsDNA, deoxyribonucleic acid, which is 50 micrometer. So this head is the genetic material DNA, dsDNA, which is surrounded by a hexagonal head. A protein and a nucleic acid content present inside the head. Nucleic acid is surrounded by a protein coat, and it's followed by, head is followed by tail which is connected by a collar and tail fibers and the basal plate. This is the structure of bacteriophage. Now life cycle of bacteriophage. When you see the life cycle of bacteriophage, there are two different types of life cycle. Bacteriophages shows two different types of life cycle. It multiplies in two different ways. They are named as lytic cycle and lysogenic cycle. Lytic cycle and lysogenic cycle. Lytic cycle is otherwise called virulent cycle. Lysogenic cycle is called as avirulent. What's the difference between these two? In lytic cycle at the end of this process, it lyses. Lysis taking place in the host. It kills the lytic cycle. The host is killed by this virus. So the bacteriophage which kills the bacteria, that is the host. But lysogenic cycle, lytic is not taking place. That's the difference among these two. Let's see one by one. First cycle is called as lytic cycle or virulent cycle. These are the diagrams which shows lytic cycle. There are 5 different steps involved in this. The first step of lytic cycle is called as adsorption. Second step is penetration. Third step is, first one adsorption, penetration, then the third step is synthesis, and the fourth step is assembly or maturation or assembly, and the last step is release of progeny, release. So these all are comes under lytic cycle. So lytic cycle, it completely lysis taking place, kills the host. So first step is adsorption. How this enter into the host, this bacteriophage, how it enter into the host and multiplies within its host body. So first step is adsorption. Adsorption is the process. Look at this diagram. This is the bacteria. Here the host is bacteria. What are bacteriophages? Bacteriophages, which viruses which infects bacteria are called as bacteriophages. So this is the bacteriophage which is going to kills the bacteria. This is the bacteria that is called as a host. This is the host. Bacteria is the host. So this bacteriophage, at first it adsorption. The first process is called as adsorption. During adsorption, there are two processes taking place. The first process during adsorption is landing and pinning. There are two different steps involved in adsorption, landing and pinning. What is this landing and pinning? At first, when this bacteriophage comes and contact with the cell wall of the bacteria, that is the host. When it comes and contact with this, it finds a recognition place or recognition site. It recognizes a particular site to sit on the, or it enter into. It finds a place, it recognizes a place. With the help of these tail fibers which attached on the surface of cell wall. That process is known as landing. The bacteriophage just lands on the surface of cell wall of bacteria. That process is landing. As soon as this landing process gets over, this bacteriophage, it starts to already there is a recognition site. In the recognition site, with the help of this basal plate, these tail fibers which bends down. So with the help of these tail fibers, here it bends down. It bends down and anchored the basal plate here. So just it anchored the basal plate with the help of tail fibers. So it bends down and it pins. The process is called as pinning. So adsorption, there are two different processes, landing and pinning. Each one is 2 mark question. What is landing? What is pinning? Next step is penetration. So here the nuclear content, the DNA material has to enter inside. It has to inject the DNA content into the host. So second step is penetration. How it penetrates. So as soon as it landing and pinning gets over, this part of here is the penetration. During penetration, the cell wall of the host started to degrade or disintegrate with mechanical or enzymatic method. It degraded. The cell wall has to degrade. Host cell wall is started to degrade with the help of the enzyme lysozyme. So lysozyme enzyme which degrades this, and this DNA content is inject through this region at the degenerated part of the cell wall. This basal plate now enlarges. The basal plate of bacteriophage enlarges and allows way to enter into the DNA content inside the host. As soon as this enter inside, now this process is called as transfection, transfer of this DNA. The bacteriophage DNA transferred into the host cell through this cell wall. So, this process is transfection. The next step is synthesis. As soon as the genetic material is transferred, when it is entered into the host, now see, when it is entered inside, it contains the DNA inside the head. Now the DNA part is entered inside the host. Now the empty shell is outside. So this empty shell is known as ghost. So when you write about this penetration, there are two processes: transfection and ghost. Two more question is there. What is transfection and what is ghost? After injecting this DNA content, this outer side of the head part is empty without nucleic acid. Now this process is known as, this part is known as the empty shell. Outside is called as ghost. So nucleic acid only, it is virulent. So this nucleic acid injected inside. Nucleic acid is injected inside, called transfection. Now the empty shell outside is called as ghost. Now third step is called as synthesis. What are synthesized? Now when the nucleic acid or the DNA, the DNA when it is entered into the host, it arrests the synthesis of host DNA, because the host DNA which started to degenerate, and the phage DNA which started to multiply. So it synthesized the DNA, the head part, the DNA part, the tail part, the tail fibers, collar, each and every part are synthesized, which is synthesized. And when the phage DNA is started to synthesize, this one started to degenerate. The host DNA material started to degenerate. This is called synthesis. And everything is highly synthesized within the cell wall of bacteria, but the nuclear content of the bacteria disintegrate. Then next process is assembly. So during assembly, the process of assembly, at first the head, tail, DNA and the tail fiber, pins, basal plate, everything synthesized separately. Now all this separated, this synthesized different parts are assembled together. Now this process is known as maturation. So at the process of assembling the head is enclosed with the DNA content which enter into the head, and the tail attached with the head connected with the collar. This process, it assembling taking place. This process of assembling is known as maturation. So now in this assembly you write about the process maturation. Two more question, what is meant by maturation? These all parts, separate parts. At first the parts are separated, the head, tail, everything synthesized separately. During the stage of assembly, all these separated parts are connected together and it assembled together. The process is known as maturation. Then final stage of this lytic cycle, the name of the cycle is lytic cycle. So at this stage it ruptures the host cell wall, because now host DNA content everything arrested. It lytic taking place, lysis taking place. It inactivates the host body. Now all these virulent progenies, bacteriophage progenies, are released by the rupturing of the cell wall. The bacterial cell wall, this is the bacterial cell wall. It ruptures the bacterial cell wall, and the phages or the bacteriophages progenies are released out by rupturing the cell wall of the host. This process is known as release of new phage particle. So at the end of this process, completely the bacteria, the host is inactivate, and the bacteriophage which multiplies and comes out. So this is lytic cycle. So very important, 5 more question. Another cycle is there, lysogenic cycle, otherwise known as avirulent. Here what happens, here these diagrams which represents this lysogenic cycle. Here what happens, the bacteriophage which attaches, at first it attaches on the surface of the cell wall of bacteria. So bacteriophage, what is bacteriophage? Viruses infect bacteria are called bacteriophage. So this bacteriophage attach on the surface of cell wall of bacteria. Bacteria is the host. So bacteria contains a nucleic acid. There is a nuclear material, and this also contains DNA. Here what happens, when this linear DNA, the phage DNA is linear. The linear phage DNA, see this is the linear phage DNA. When it enters inside this host DNA, it becomes circular. So linear DNA, bacteriophage DNA is linear. When it enters inside, it becomes circular DNA, circular phage DNA. This is the circular phage DNA and this is the bacterial genome, bacterial DNA content. This genetic material of bacteria. This is genetic material of bacteria. This is the integrated phage DNA. Now both the DNAs are present within the host cell. Next phase, the next stage is, once this bacteriophage DNA enters inside the bacterial DNA, which integrates together by the process of recombination, which combines together. So this part of the bacterial DNA and part of the bacteriophage DNA integrated together, and now this stage is called as prophage. So now this phage DNA is known as prophage. A new DNA, recombinant DNA is formed. So it is integrated. This completely not kills the content of DNA content of the host, but both are interconnected. It interact together and forms a prophage. So this prophage is represented by, two repressor proteins to control over this synthesis of progeny. So this prophage is synthesized. So this prophage, when the bacteria multiplies, when this bacterial cell reproducing, the prophage multiplies. The prophage multiplies within the host, and when the cell divides, the prophage is multiplies. So both the genomes or the genetic material both mix together, and it cannot completely kill the host. That's the difference between the lytic cycle and the lysogenic. When you compare this lytic cycle, the bacterial genetic material will not be there. Instead, the bacteriophage DNA completely synthesize and release the progeny. But here there is a integration, and that integrated the DNA content is called as prophage. So prophage multiplies whenever this bacteria reproduces. This cycle is called as lysogenic cycle. Hope you understand clear. So today what are the questions? You have to learn first definition, define bacteriophage and structure of T4 bacteriophage. Draw this diagram. This is another diagram. Lytic cycle, life cycle of bacteriophage. Two types, lytic cycle, lysogenic cycle. So this is one diagram, this is another diagram, this is three different diagrams with different questions. This is 5 mark question, this is also 5 mark question, structure of T4 bacteria 5 mark question, and define bacteriophages 2 mark question. Before the next class, learn everything. Thank you.